Good morning, good afternoon, and good day to everyone. My name is Namali Jaya from CGen, and along with Tara Enda, I'm one of the co-chairs for this year's CE Pharm. It is my greatest pleasure and privilege to welcome you all to another exciting, information-packed program on capillary electrophoresis. It is so great to see so many familiar names, if not faces, and along with some new names that I'm really looking forward to getting to know better during the next 4 days. So let's start with a few reminders. Please make sure your system is appropriately working by running all the checks and that your sound is working and you can hear me. This is our second virtual CE Pharm, and we're getting the hang of virtual networking. But please make sure you be intentional about that networking. If you haven't already, please connect with your fellow attendees via the chat function that you see on the left-hand side of your screen. You can use the Q&A feature to ask questions. And finally, don't forget to opt into the attendee list to share and connect with others. I checked in and there's about a list of 15 and growing. So please go in and opt in to share information with your fellow attendees. A little bit about CAS. If you're new to the CAS family, CAS is a nonprofit scientific organization who excels at bringing professionals from industry, academia, and regulatory agencies together to solve challenging technical issues and share information. Another one of my favorite aspects of CAS is the intentional commitment to diversity, equity, and inclusion. This is upheld by the CAS Board of Directors because diverse perspectives really drive innovative thinking and better solutions. As we're doing today, CAS brings communities together for knowledge sharing. And there are multiple symposia that are in place to carry out this mission of knowledge sharing, along with CE Pharm, which I love because this is like coming home to a family reunion. My favorite CAS meetings are WCBP and CMC strategy forums. I have also attended numerous regional forums and always rewarded with meeting great people and learning new topics and finding answers to problems I may have had. I also want to make a plug to this CAS mass spectrometry forum happening next week. So if you're interested and the CE forum has whetted your appetite, please sign up for that as well. CAS is truly a global organization with participants from around the world. While most of our attendees are from North America and Europe, we're continually growing our footprint in Latin America, Europe, and APAC regions. As shown here, the meetings are attended by industry, regulatory, and academic colleagues, and we work to facilitate industry and regulatory interactions, as well as agency-to-agency dialogue, which is really instrumental to driving our strategies forward. And really, it takes a village to develop the content of these forums. This is our organizing committee who are genuine CE enthusiasts. These people are fantastic. They are responsible for the great program you will be experiencing in the next few days. If you're interested in participating in the organizing committee, please indicate that in the survey at the end of the conference. We're always looking for new members and fresh perspective. And of course, our village extends to the small but mighty CAS staff who work alongside and behind the scenes tirelessly to make this forum a success. These are the names of those fantastic folks, and thank you to all, and a special shout out to Julie Harris. She has been alongside myself, Tara, and the committee putting this program forward. So thank you to all of these fantastic people. And a special thank you to our program partners whose support is greatly needed and appreciated. This great list of companies are here and have funded us to make this program possible. and the list is growing. So if your company is interested in funding us, please let us know, and we can include your name up here as well. And we love our vendors, because without them, really, where would we be, right? Because they develop new technologies which really move our therapeutics forward. Please take the time to visit the vendor booths and attend the partner showcase and technical seminars to learn more about the new technology advances. Thank you, thank you, thank you to all of our vendors. As I said earlier, please opt in to take the most advantage of this virtual forum and add your name to the attendee list so that we can reach out to you. Also this year, we have our poster session and the troubleshooting workshop, what we fondly call the Tim and Carrie Show on Wednesday. They will be on the Remo platform, along with our roundtables and the CE Pharm presentation. So make sure you also go into the Remo platform and that you have the proper access in there as well. So take the time to set that up. And again, as I said, we love our vendors and they have booths set up. So please make sure to visit the vendor exhibits and chat with them as well, because they're always prepared to partner with us in solving our technical problems. Finally, we have a virtual poster session, which is promised to be fantastic as well. Make sure you join that, and there's an opportunity to talk with other attendees as well as the poster presenters themselves. I'm really looking forward to the poster session this year. As I said earlier, we have the virtual roundtables that are occurring in Remo. This is, again, there's a lot of aspects of the CE Pharm that I love, but the roundtables are always near and dear to me because it allows me an opportunity to troubleshoot some of the hot topics that might be front and center in my mind and learn from others. So please attend the roundtables that are being held on Wednesday. Finally, once again, I just want to remember to use all the tools available to you. And most importantly, enjoy this virtual CE forum. With that, without much further ado, I want to hand it over to Nate to introduce our keynote speaker. Nate?
Hello, everyone. I am Nate Locker from Pfizer. I'd like to first start by thanking you all for joining us at the 2021 CE Pharm Virtual Symposium. It is my pleasure today to introduce our keynote speaker, Professor Susan Lunte, from the University of Kansas. So Sue is the Ralph N. Adams Distinguished Professor of Chemistry and Pharmaceutical Chemistry at the University of Kansas. She received her bachelor's in chemistry from Kalamazoo College and her PhD in analytical chemistry from Purdue University. Over her time, she's hosted visiting scientists from all over the world, had greater than 20 postdocs, almost 60 graduate students, several undergrads doing research, over 165 publications, 21 book chapters, is on the editorial board of many prominent journals, and has been on scientific committees and corporate organizations. Her areas of specialization include bioanalytical chemistry, liquid chromatography, capillary electrophoresis, electrochemical and laser-induced fluorescence detection, microdialysis sampling, neurochemistry, protein and peptide analysis, microchip analytical systems, mass spectrometry, and anti-cancer drug analysis. The interests of the Lunte group include microanalytical methods for the investigation of transport and metabolism of peptides across the blood-brain barrier, separation-based sensors employing online microdialysis coupled to microchip electrophoresis, cell-based assays on chips, and microchip-based diagnostics for cardiovascular and metabolic diseases. So, it's really my honor to introduce her today. I am one of her former students. As I mentioned, she's had a lot of students over the years that are probably working with you now. So I'm guessing that some of the knowledge that she has given to her students has probably been transferred, to you through your working experiences. But again, it's my pleasure to introduce Sue, and I will hand the floor over now.
Hey, thank you, Nate. It's always great to have one of my more successful students introduce me at a conference, and I appreciate the invitation to be a keynote lecturer. I really have enjoyed this meeting in the past, and it'd be really great if when it's in person again, where we can actually all see each other and have a beer or whatever. So what I wanted to talk today about is some work that we've been doing in our lab over the past 10 years using capillary and microchip electrophoresis to look at biomarkers of oxidative stress. And as you know, Alzheimer's is a devastating disease that affects around 5 million Americans each year. And nitrated, it's been found that nitrated proteins are present in high concentrations in the brains of Alzheimer's patients that indicate the presence of these reactive oxygen and nitrogen species. So one of the things that we're interested in is looking at trying to develop analytical methods to look at different substances that are involved in neuroinflammation and small biomarkers that can be used to monitor oxidative stress. So this includes nitric oxide, peroxynitrite, nitrate and nitrite, which are formed from the dismutation of peroxynitrite, and also look at glutathione and other antioxidants that are present in the cell to detoxify some of these reactive oxygen and nitrogen species. So the overall goal has been to develop microfluidic, including capillary electrophoresis methods, to look at all the species that we can that are implicated in the evolution of oxidative stress. So like I said, nitric oxide, nitrite, peroxynitrite, and then also look at some products of protein oxidation and nitration as well. So what's one of the big analytical challenges with the reactive oxygen and nitrogen species is that they are reactive, and they only have very short half-lives in vivo, usually less than a few seconds. And there are actually methods that can detect these species. There's electrochemical methods, but they're generally specific for one particular analyte. The Amatori group in France has done a lot of work where they've been able to detect directly peroxynitrite. But there's also sensors for nitric oxide, but these are usually specific for a single analyte. And also there are fluorescent probes to detect some of these species, but they are also tailored to just detect one specific type of reactive nitrogen and oxygen species. So what our approach is to use a separations-based approach so that we can look at multiple species simultaneously. And so that's where the capillary electrophoresis and microchip electrophoresis comes into play. So actually, Nate was one of the first people to do microchip electrophoresis in my lab many years ago. And we found that it has a lot of advantages for bioanalysis. In particular, for reactive nitrogen and oxygen species, the fast separations allow you to detect these species before they degrade. So like I said, they only last for a few seconds at physiological pH. And if you can do a separation, a sub-minute separation, the chances of actually catching these is much higher. Also, we can analyze small sample volumes, and I'll show some examples where we use this technique to analyze single cells and also microdialysis samples. The small footprint makes it easy to move around the lab. And with COVID, I've been thinking about sending these instruments home with people so they can actually do it next to their coffee maker so we can get more work done. Also, there's a potential for integration, and you can use a variety of detection mechanisms. The ones I'll talk about today are mainly electrochemistry and fluorescence, but it's also compatible with mass spectrometry, conductivity detection, and other variety of detectors. So specifically for the small molecules, the small reactive nitrogen and oxygen species, like I said, the microchip electrophoresis allows you to actually monitor multiple species simultaneously, and also to be able to detect these very short-lived species, things that only last a few seconds. In addition, because there's no stationary phase, you can use high pH buffers, or you can add strange additives to the capillary without having to worry about destroying stationary phase. And then you can also, if you use electrochemistry, you can do direct detection of some of these analytes. And in fluorescence, you can do indirect detection. And then, as I said, it's possible to also apply this using single cell analysis. So as I said, Nate was actually one of the pioneers of electrochemical detection in my lab on microchips many years ago. And electrochemical detection is great for microchip separations because, and it's particularly useful for detection of reactive nitrogen and oxygen species because oxidative stress, the products and reactants of oxidative stress are almost by definition electrochemically active because they're redox reactions. So the things that are produced and the things that are oxidizing are generally either oxidizable or reducible. So EChem detection is like a selective detector for these types of compounds. In addition, you can easily integrate by using the same methodology that you use to make the microfluidic chip, you can integrate the electrode into the chip. And then I'll show in a little bit some different electrode configurations can be used that you can use to either get selective detection of specific compounds or do voltammetric characterization, which is like dual wavelength monitoring to actually get information, more conclusive identification about the peak that you're interested in. So the analytes that we've been interested in using the electrochemical detection are the reactive nitrogen oxygen species and the antioxidants that are found in cells. And then we've also been trying to look at modified amino acids and proteins. So tyrosine isomers that result from phenylalanine oxidation, dityrosine, L-DOPA, and nitrotyrosine, which is a reaction of tyrosine with peroxynitrite, and chlorotyrosine, which is with hyperchlorous acid. So just to show one example of a fast reaction product that can be detected using this microchip, here we have, this is sort of an old example, but we have proline nanoate, which produces nitric oxide. And you can actually use the microchip to separate out the proline nanoate, which is the parent compound, from nitric oxide, which is being produced, and then nitrite, which is being generated as the nitric oxide dismutates. The other thing is the nitric oxide also is a gas, so it leaves the sample well, so it continues to go down. But you can see we can separate the nitrite, the proline nanoate, and the nitric oxide using this microchip in less than 30 seconds. We can also separate and detect peroxynitrite. So if we use reverse polarity on a microfluidic chip and then make it at a very high pH, and so that the peroxynitrite is an anionic form, which is a stable form of peroxynitrite, we can actually separate and detect the peroxynitrite and separate it from nitrite in this particular sample. So this was a commercially available sample from Cayman Chemicals, and you can see that we can separate out these compounds. So then we wanted to, so we can detect these things, but then we wanted to see, can we actually detect these in cells that produce these reactive oxygen and nitrogen species? So we chose macrophages as a model system. They are easily activated by lipopolysaccharide or polymerase acetyl acetate to produce nitric oxide and superoxide. And so the idea was to try and induce these different pathways in these cells and then look for the products using the microchip electrophoresis with both electrochemical and fluorescence detection. And with our separations-based method, we can look at a number of different species simultaneously, including pro-oxidants and antioxidants. So as I said, but we used just raw 254 macrophages to do these studies. And basically we have, in the studies I'm going to show right now, we only induce the nitric oxide pathway by adding lipopolysaccharide, which makes the macrophage think there's a bacteria around, and then it induces nitric oxide synthase, producing nitric oxide. And so here we're just trying to detect NO or its degradation products, nitrate and nitrite. So to do these studies, we grew these macrophages, we exposed them to LPS, and you can see once they're exposed to LPS, then they start forming processes and start generating nitric oxide. And so then we harvest the cells, centrifuge them very quickly, lyse them and buffer, and then immediately inject them into the microchip. And if we do this, we can actually catch the nitric oxide that's produced. Actually, this is the NO peak here. Actually catch the nitric oxide that's produced by these macrophages in that lysate. But of course, nitric oxide being a gas, it doesn't last for very long. So alternatively, we can look at some of the other RNOS markers in the cell. So this shows a number of different compounds that are found in the macrophage lysates, including nitrite, which is an indicator of nitric oxide production, glutathione and ascorbate, which are major antioxidants in the cells, tyrosine, which is an endogenous amino acid, and then hydrogen peroxide, which could also either be in the peroxynitrite sample or potentially in the cells. So just to show an example, if you don't hurry and you let the analyze the sample and allow the nitric oxide to be converted into nitrite, you can see again that the LPS stimulated cells have more nitrite than the native cells, and then you can also detect the glutathione that's in the cells. Now, since nitric oxide is not a pro-oxidant, not until it reacts with superoxide, we were interested to see if any of the glutathione had been oxidized to glutathione disulfide. And if you look at this, what you see is that really we're just seeing the formation of nitric oxide, but the amount of glutathione between the stimulated and non-stimulated cells isn't changed. So until we actually induce peroxynitrite, we probably won't get this increase in or decrease in glutathione. Whoops. Another area that we've been looking at is looking at amino acid modifications in proteins by these reactive nitrogen and oxygen species. And Danushka in my lab has been looking at the reaction of phenylalanine with the Fenton reaction to look at the formation of meta and orthotyrosine, and also looking at the reaction of paratyrosine with hydroxy radical to form L-DOPA, as well as dityrosine and then with peroxynitrite to form nitrotyrosine. And so these were the compounds that he was really interested in, and I thought it might be interested in this group because he's using a sort of an interesting separation method to do this. He's using ligand exchange micellular electrochemical chromatography. So with this technique, he adds copper and hydroxyproline to the run buffer, and this hydroxyproline is complex to the copper, but when tyrosine is injected, it replaces the hydroxyproline, leading to a change of mobility for the tyrosine. And then he also includes a micellular electrokinetic component to help resolve some of the compounds. So if you look at this separation by CE, you can see you get a pretty nice separation of all the compounds that I was talking about, L-DOPA, all three isomers of tyrosine and also nitrotyrosine. The other thing you notice is that it takes 20 minutes to get all these things separated. And also we have this sort of weird baseline thing going on around 13.5 minutes. But Dulan's worked on this and he's gotten, or not, sorry, Danushka's worked on this and he's gotten its MEEC separation of the same compounds. And this is very interesting because, first of all, it's much faster. There's a lot of lead time here, but it's still less than 2 minutes. And you detect all the same compounds, L-DOPA, P-tyrosine, metatyrosine, orthotyrosine, and nitrotyrosine and dityrosine. The interesting thing is he's actually also getting a chiral separation of the three of the metatyrosine and orthotyrosine. So the isomers or the standards that he was using were chiral, so he's actually getting a separation of the two enantiomers. So then we're interested in, just to demonstrate how this works, just taking the Fenton reaction, adding the phenylalanine to the Fenton reaction, and then analyzing it by the by the capillary or by the microchip electrophoresis method that he had developed. And so here we add iron and hydrogen peroxide in the presence of phenylalanine to form the hydroxy radical, then react it at 1 hour at room temperature, and then dilute it quickly with background electrolyte and run the electrophoresis. And so if we do this, you can see that he does actually get all 3 tyrosine isomers as products of the Fenton reaction. And so he's now applying this to the tyrosine as well to see if he can get, when he does tyrosine, he does get the dopamine or L-DOPA, but he hasn't yet seen the dityrosine. So we're now working to see if we can look at that Fenton reaction product as well as a reaction with peroxynitrite. So another area of reactions of tyrosine with different amino acids or with different reactive oxygen and nitrogen species was something that Kelsey Shilley worked on for her PhD thesis. And I think she gave a presentation on this, some of this work last year. But so with her work, she's been looking at mainly the formation of nitrotyrosine and the different pathways to form nitrotyrosine from peroxynitrite formation. And so peroxynitrite can be synthesized in the lab by a method that Joe Beckman put together a while ago. And what you do is you take nitrite and hydrogen peroxide in a very acidic solution. And when you put those together, you start getting peroxynitrite. But if you have to quench it very quickly in base in order to stabilize the peroxynitrite, or it all degrades to nitrate and nitrite. So if you take longer than a second to add the sodium hydroxide, you basically lose all the peroxynitrite. So this is just the assays on a spectrophotometer, so you can see with 1 second reaction time, you get a lot of peroxynitrite, which has a unique absorbance at 302 nanometers. But if you wait for 5 seconds, all you get is absorbance for all the products, the byproducts of the peroxynitrite degradation. So you can also analyze your reaction mixture with CEUV. It's very helpful for peroxynitrite. So if you do reverse polarity CEUV, you can separate out nitrite and nitrate and hydrogen peroxide and peroxynitrite at 214. You can see all of them. If you want to see which peak is peroxynitrite, you can just dial in at 302 and you can see that there's a peak for peroxynitrite. This is really actually a useful assay because we get a lot of commercial. Now we're making our own peroxynitrite, but when we were getting commercially available peroxynitrite, as I'll show a little bit later, we'd frequently find out that it really didn't have any peroxynitrite in it at all. It was just nitrate, nitrite, and hydrogen peroxide. So the ability to actually analyze the samples with a separations-based method as opposed to a UV spectrometer really gives you more information about what's in those things that you're getting in the mail. So one of the things that she looked at, she and a very talented undergrad, Riley Stegmeyer, looked at was the reaction of peroxynitrite with tyrosine and the difference in degradation time. So if she reacts the tyrosine directly within a second with the peroxynitrite, then what she'll find is that she gets the nitrotyrosine formation. But if she waits, if she makes this degradation time like 5 seconds or 10 seconds, then there's no reaction with the tyrosine. If she waits 10 seconds to add the tyrosine, there's no product. And so you can see that here. These are standards just separated by CEUV. And then And then if you add the tyrosine within 1 second, you can see that you form the nitrotyrosine. But if you if you wait for 10 seconds to add the tyrosine, then all you get is tyrosine. So you have to really be able to add the tyrosine quick enough so that it actually reacts with the peroxynitrite that's been formed before it degrades. And if you look at sort of it's not really the kinetics, but if you look at a plot of degradation times versus normal peak height, you can see that you really need to, the peroxynitrite is really degrading. And if you don't add it within 1 or 2 seconds, mainly you just get whatever you added, in this case tyrosine, and very little product. So the maximum amount of nitrotyrosine that you get is really if you add the tyrosine within 2 seconds. So this led us to looking at some peptides that the Schoeneck group here at KU were interested in. So they've been looking at the role of these nitrated peptides in Alzheimer's disease, and they've been using phospholamban as a model protein that's highly nitrated. And so there's several people in his and John Stovall's group that have looked at methods to analyze these nitrated peptides. And so we wanted to see if we could separate out these peptides and their nitrated products using UV detection. And so these include, of course, we had the tyrosine and and then we have AY, AYL, and then this larger peptide, F Salar. So these are all parts of the protein that are nitrated in vivo. We also, Kelsey was also interested in looking at some other potential products, in particular chlorotyrosine, that's sometimes found in vivo, and then aminotyrosine, which is the reduced form of the nitrotyrosine, and is also electrochemically active and can be tagged with some agents. So she and Riley developed this really nice CE separation of all these different peptides, and it's interesting because it's a pretty simple separation buffer. And you can see that all the nitrated peptides have much lower electrophoretic mobilities than their parent peptides, so they all migrate later. But they're easily separated from their parent compounds. So she has this nice separation of all the peptides involved, or not all, but some of the key peptides involved in this protein nitration. And so they did the same sort of assay where they did 1 second degradation time, or 1 second time before they added the analyte, or 10 seconds, to see if they got the same sort of response that they got for the tyrosine tyrosine itself. And you can see here's the separation of all the peptides. Again, if you do the reaction quickly, you get mostly nitrotyrosine. If you don't do it quickly, you get mostly the parent compound. But the same thing holds for the dipeptide and the tripeptide. Whoops. She wasn't able to separate the F Salar one from the product, but you can see, you can definitely see that the peroxynitrite is reacting with each of these peptides. So then we tried this on chip, and we have a separation of like the parent compound and a nitrated compound for each of these peptides. But unfortunately, we haven't been able to yet separate all the peptides using the Microchip CE. But this is with electrochemical detection. So we're able to not only detect these with CEUV, but also we can detect them electrochemically using a 5 centimeter channel and a potential of about 1 volt. So that led us to this other idea that we had, is that can we selectively detect nitrated peptides in a complex mixture? And so I'm going to just talk briefly about each of these, but you can use this dual series mode to selectively oxidize something at the first electrode, and then oxidize or reduce something at the first electrode, and then reduce it at the second electrode. And then we can also use this dual parallel one, which I'll talk about briefly, to voltammetrically characterize what's in a sample. So why would we want to do this dual series electrode? Well, we had the idea with nitrotyrosine. Nitrotyrosine is reducible, and there aren't very many reducible things in biological samples, but reducible things are hard to analyze because of oxygen interference. So the idea was, if we could reduce the nitrotyrosine at the first electrode, and then reoxidize the oxidation product at the second electrode, that would give us a sort of a selective detector for nitrotyrosine containing peptides. So the idea here, this is a cyclic voltammogram, but the reaction is reversible, so you can reduce the nitrotyrosine to the hydroxylamine and then reoxidize it to the nitrosamine at these two electrodes. And so you can see that you get this reversible CV here in the cyclic voltammogram. And so this is some of the data she got. So this is actually a mixture of tyrosine, which is not reducible, and 3-nitrotyrosine, which is reducible. So at the first electrode, we have it set at minus 200 millivolts, which is a reductive potential. So it's going to reduce nitrotyrosine to 3-aminotyrosine or to the hydroxylamine. And then if we set the second electrode at 600 millivolts, it's not high enough to detect the tyrosine, but it's but we do get the oxidation of the product of the reduction of the nitrotyrosine. And this works also for the peptides. So if we take the dipeptide, the nitro dipeptide, we also see that it's oxidized at the second electrode. So there's some hope that this could be used to selectively identify nitrated peptides in a mixture of a lot of other peptides. The other thing that you can use electrochemical detection for is voltammetric characterization. And Dulan Gunasakara in my group developed this dual pair electrode configuration that looks like a weird looking spider or something, or something from some cartoon. But anyways, it's got 2 sample wells. And basically what you can have is 2 separation channels. And you can so the way this works is it injects equal amounts into both of these channels. And then if you set these 2 electrodes at 2 different potentials, then you can actually get voltammetric characterization, which is similar to dual wavelength monitoring to look at peak purity. So you can get a sort of an idea of the identity of a peak, even if you're only injecting a few picoliters. And the one crucial thing here, which Dulan had great eyes, is actually trying to put these electrodes at exactly the same spot at the bottom of the channels. So first thing you want to do is make sure that the 2 electrodes are aligned and that they're giving approximately the same response if the same potential is applied to both electrodes. And so this shows that that's pretty much the case. If we apply 1100 millivolts to both electrodes, we get pretty much the same response, a ratio of 1 for all these different compounds. So that shows that the electrodes are aligned and that we're getting the same amount injected in the 2 channels. So here's a standard that we did get from Cayman Chemicals that had known peroxynitride in it, and just to show that this can be used to identify impurities in samples. We ran this sample at 1.1 volts at 1 electrode and 0.9 volts at a second electrode. So we're looking at the current ratio as a method of identification. And you can see that nitrite, peak 1 is pretty much nitrite, but peak 2 is hydrogen peroxide. It's not peroxynitrite as advertised, and there's no peak for peroxynitrite, which would be more easy to oxidize than this. We also applied this to our macrophage lysate that I showed earlier. And so here's the macrophage lysate at 1100 millivolts, and here's one at 950 millivolts. So, so peak 1 is definitely nitrite based on the current ratios, but we need to probably pick a better potential. These are close, but it might be better if we had, like, the best thing to do is get it on the curve part of the electrochemical response, so you get the best current ratio. And so we might need to pick an additional potential there. Whoops. And then, but this last one is definitely not hydrogen peroxide, and we think it's probably nitric oxide. So that sort of talked a little bit about what we've done with electrochemical detection, and I just want to briefly talk about what we've done with fluorescence as well. So just like, so we can separate multiple species using electrochemical detection, or using microchip electrophoresis with electrochemical detection. Although dyes are specific for specific probes, a lot of them are not, have side products that show background or give false results for the assay. So one example of these is DAF-FM, which is used to to monitor nitric oxide. And so DAF-FM enters the cells. So there's an esterase that enters the cells, and that reacts with nitric oxide to form this DAF-FMT. In all our cell studies, we've also added CSDA, which is a non-fluorescent, is a similar compound. It's non-fluorescent, but once it enters the cells, it becomes fluorescent. So it's nice because this can be used sort of as an internal standard for both cell volume and for esterase activity in the cell. So as I mentioned, with this DAF-FM, the Sweetler group found that the DAF-FM also reacts with ascorbic acid, which is in a lot of cell samples. And so we were able to separate the product with nitric oxide from the product with the dehydroascorbic acid. And so when we did just bulk cell lysates of the cells that I showed before, the macrophage cells that were exposed to LPS, what you see is that you get a larger amount of DAF-FMT with the LPS stimulated cells, but we also, I mean, it's not a lot here, but we did not get an additional, we didn't read additional fluorescence due to the reaction with the ascorbate. We also have looked at mitosox, which is used a lot for monitoring superoxide. And so the product is this 2OH mito-E. And if you look at the fluorescence of these compounds, the mito-E is fluorescent. And so if you just take superoxide produced from xanthine oxidase and react it with mitosox, you'll get 2 peaks. If you separate them by microchip electrophoresis with fluorescence detection, you'll get one for the reagent and another one for the product. If we add superoxide dismutase, which eliminates the superoxide from the xanthine oxidase, then we'll just get the product, the mito-E product. And so here we stimulated the macrophages with polymericytyl acetate, which causes the formation of superoxide. And so here you get the formation of the product, and then we have the 6CF as an internal standard again, and you can see the native we're not seeing as much. And then Giuseppe, who was working in my lab at the time, decided, well, let's try both. So he added both DAF-FM and mitosox sequentially to stimulated cells to see if we could actually monitor both of them in the same run. And so here we have the microchip electrophoresis with LIF detection of both nitric oxide and superoxide. So here, this is the native cells, so there's really not much in there. You can see it's quite noisy. If I add SOD inhibitors to reduce the amount of, to eliminate superoxide dismutase, removing the superoxide from the cell, then we get a buildup in the cell, and you can see you get an increase in the 2-hydroxy mito-E and a little bit of the DAF-FMT. And then if I stimulate the cells, you can see we get increases in all of them, and we still have this DAF-FM DHA peak in there as well. So this just shows the ratio of nitric oxide to superoxide for the different, under the different stimulation protocols. So it's just sort of an aside. We sort of got interested in this dipeptide carnosine. People that are into bodybuilding may know about this. It's an anti-aging nutritional supplement that has antioxidant properties. You can get it at natural products, and it's been thought to help with reactive oxygen species and reactive nitrogen species detoxification. So we wanted to see, well, can we use these methods to see what carnosine does to these cells? So basically we added carnosine. We had the native cells stimulated with carnosine, or the stimulated carnosine, and then actually this should be native plus carnosine, and then stimulated with adding carnosine. So the first thing that was interesting is because CE is so good at analyzing amino acids, we could actually, this is just this is with microchip electrophoresis with LIF detection using a microchip, but we were able to separate out like the intercellular amino acids of the cells that we were analyzing before we did these experiments with carnosine. So we wanted to see if there's any endogenous carnosine in there at first. And so the endogenous, there's a peak that migrates with carnosine, and when you spike it, it comes out at the same time, but you can tell there's very little endogenous carnosine in the cell. However, when we add carnosine to the media, the cell uptakes these, or the macrophages uptake these carnosine quite a bit. And the more they're stimulated, the more they take up. So we found that was pretty interesting, that stimulating the cells actually increased the amount of carnosine that they brought in. So the other thing that was interesting is that when he did the CELIF or the MELIF analysis of the carnosine, the effect of carnosine on nitric oxide production, you can see that it completely knocks it down compared to what you would see if you don't have carnosine. So it's definitely either inhibiting the nitric oxide synthase or somehow trapping the nitric oxide. Also, he looked at superoxide production, and you can see it's not quite as dramatic, but it also seems to reduce the superoxide production as well. But it does seem to have a dramatic decrease in the amount of nitric oxide. So we're still exploring this. So one of the ultimate goals of all this is to take all these fluorescent probes and also the electrochemical detection and be able to do analysis cell by cell. So we've been working with Chris Culbertson on a single cell analysis system. And the idea here is to be able to bring a cell into the system. The high voltage that we use for the separation will immediately lyse the cell, and then we can separate out the contents of the cell in the separation channel. And because you can dilute the cells so they just sort of march in here one by one, you can do multiple individual cells quickly in a quick fashion. So this is sort of an old slide, but it shows how this works. So you bring the cells in, they enter the channel, they're lysed, and then the cell debris goes through this channel, the big stuff, and then the small molecules that we're interested in go into the separation channel. So this just shows a single Jurkat cell that we did. So here, again, we have this CFDA label in there as a marker. And then the DAF-MT tells us how much nitric oxide is. We actually lyse it so quickly that we get both hydrolysis products of CFDA. So we actually have two different peaks for that. But if we take the peak height ratio of the DAF-FM to the CF peaks, what we see is that we get a very interesting distribution. So the stimulated distribution is much wider, but the native distribution is is narrower. And the other thing that's really interesting is if we take the single cell analysis, we took 100 cells and determine what the ratio of the DAF-FM to 6F is, and then we take the same, compare it to the bulk cell analysis where we take a whole bunch of cells and do the same measurement, you can see that they come out very close. So both of them show about a 2.2-fold increase in the amount of nitric oxide that's produced. So Chris has been working with us to do more high throughput analysis, and now he's up to, I think he might even be higher than this now, 15 cells per minute. So the idea is to take our assay where we could do superoxide and nitric oxide, and hopefully also a probe for peroxynitrite, and develop an assay where we could run multiple single cells with multiple markers in them to look at all the different oxidative stress markers. Okay, so the last thing I want to talk about, and I heard that somebody at least wanted to hear about this thing, is to transfer this more into in vivo analysis. And one of the ideas that we've had is trying to do near animal or near patient or on animal sort of separation-based sensors for these reactive oxygen and nitrogen species, and also other indicators of oxidative stress. So the idea is to use this technique called microdialysis sampling as sort of a generic sampling method, and then separate out the compounds that are sampled using microdialysis sampling using microchip electrophoresis, and then detect the products. In this case, I'll show right now is with electrochemical detection, but it can also be done with fluorescent and other types of detectors. So microdialysis sampling is a really great thing to couple to these chips because it acts like a synthetic blood vessel. Basically, what you do is you pump a fluid that's similar in ionic strength to whatever tissue you're trying to interrogate. So if it's the brain, it's artificial cerebral spinal fluid, or you can use Ringer solution for blood. And the nice thing about the microdialysis is it's an online sample preparation technique as well. So only small molecules will go, it's basically a dialysis membrane. So only small molecules will go across the probe, and they will move across the probe based on their concentration gradient. So molecules that are in extracellular fluid that you're interrogating will go across this probe, and then they'll be pumped slowly to the analysis system. So we're pumping perfusate slowly through this probe. Analyte that's in the extracellular space will move into the probe and then be pumped to the analysis system. The other thing that's interesting about this technique is, one, proteins are excluded, but also protein-bound drugs or protein-bound chemicals will also be excluded from the membranes. They won't pass through the membrane. So if you want to measure, let's say, the free concentration of a drug, this is a good way to do this because all the bound drug will not pass through this membrane. Also, it's usually the perfusate is just a salt solution containing small molecules, so it's really amenable to injecting into a microchip or a CE system, since you don't have to do any protein precipitation or remove any cells or tissue. And the other thing that's interesting is that enzymes can also not cross this membrane. So if you want to look at, in this case, I'm looking at small molecules, but if you want to look at peptides or something, then once they cross this membrane, they're isolated from peptidases and things that can change your results because the enzymes won't cross the membrane. And lastly, because you're pumping this through this probe on a continuous basis, you can implant these probes in an animal, allow it to recover, and do continuous measurements on awake animals and also freely moving animals for several hours, in some cases even longer, because basically there's no fluid removed from the tissue. So the typical microdialysis setup is looks like this, where you have a rat in a bowl with a syringe pump, the perfusate's going into the probe, and then you have the online. Then you put it to a fraction collector or an online connector. So for our purposes, we've connected this to microchip electrophoresis by using a top area here, which where the flow goes through, the microdialysis flow, and then we use gated injection to continuously inject sample through the probe. Now, the reason that we do this, I'll show in a minute. So basically, this is a nice way to do it because it can easily be done remotely, because you're basically just changing the voltages at these two reservoirs to make the injection. So if you want to program something by Bluetooth so they can be used with Bluetooth and done remotely, this is a great way to do it. There's a lot of other ways that are really good to do it, like droplets and pneumatic valves, but they're less easy. They're not as amenable to the like on animal type of application that I'll be showing in a minute. So this is the overall system that we have. Here's the potentiostat, here's the high voltage power supply, here's the syringe pump, and here's the chip. And the animal is not actually in here yet, but the idea is then we had this idea of trying to make this portable so that we could actually do on animal experiments. And some of you, I'm sure, have already seen this. But the idea was to integrate the whole thing so that it could be on animals, so we can actually eventually do behavioral studies or other studies, drug delivery studies, neurochemical studies on animals that were freely moving and freely behaving. And we'd love to be able to do this on a rat, but so far the device is too big for a rat. So we worked with Pinnacle Technologies to develop a device that basically fits in a lunchbox and then can sit on the top of the sheep. And so, yeah, we call this our lab on a sheep project, and we have this nice little harness that Nate knows, Kathy Heppard. I think she found this. It was like a rock climbing thing for dogs, and she's like, "Oh, this would be perfect for for the sheep." So, I mean, we actually had a little sheep vest at first with the power supply in one pocket and the potentiostat in the other, but this climbing vest for mountain climbing for dogs actually worked really well, keeping the device on the sheep. And so the whole thing is remotely controlled. The sheep is well, he's not freely moving right now. He's tethered to it. I mean, he's being walked around, but the whole setup is in this Faraday cage. There's a syringe pump. The microdialysis probe for the first time we did it was just done for transdermal delivery because we didn't want to do any brain surgery till we were sure this was going to work. And so we have the whole setup in the Faraday cage, and it's all run by a laptop battery and Bluetooth. And so the first application that we did was actually looking at nitroglycerin, which produced one of our favorite metabolites, nitrite. So we perfused the nitroglycerin through the probe, went into the skin, formed nitric oxide, and then we can monitor nitrite. And so this is an animal that was at the sheep that we did these experiments at Purdue, and this sheep was in a barn, which was a real challenge because that's part of the reason we had the Faraday cage. There's a lot of noise in a drafty barn in the vet school at Purdue. But what you can see is we can actually see the production of the nitrite over time. And because nitrite was really the only major product that was getting through the microdialysis probe, we could get a really nice separation here. There is a lag time for all the tubing that goes from the different parts, but once that lag time's over, then you can detect nitrite. We also, just to make sure this was working okay, we put a second probe on a different part of the animal and then collected 10-minute samples, which is what a person would normally do, and analyzed it by HPLC. And you can see, even though it's on different parts of the sheep, we're getting pretty similar results between the two. So what we're working on now is trying to use this for catecholamine analysis so we can look at the effects of drugs on neurotransmitters and also look at behavior and neurotransmitter release. And so Rachel Saylor and my group developed the separation of catecholamines, and then Shamal and Galina have been continuing this work along with another talented undergrad, Anton. And so you can see we're trying to monitor these things continuously. And we had, we did do a rat. So we perfused with high potassium, and you're able to see, it's hard to see on this, but you're able to see that there is an increase. If you look real carefully at peaks X, you can see that there's an increase in the and this one peak upon high potassium perfusion, but we can't detect dopamine yet, endogenous dopamine yet. So that's our sort of our current goal, is to be able to detect endogenous levels. So right now we can look at the increase in dopamine due to the high potassium perfusion, and this shows that in this and that graph, but we can't actually detect the endogenous levels yet. So we're working on that so that we can actually then do some animal studies where we can continuously monitor catecholamines and then look at the effects of behavior, either social behavior or drugs on catecholamine release. So our idea on this part of the project is really to develop a modular platform where we have different chips for different types of analytes. I showed the one for nitrite and the one for catecholamines, but we're also working on some for amino acid neurotransmitters, adenosine metabolites, and other reactive nitrogen and oxygen species. And the idea would be to have custom chips for the particular application that you could just put into that device and then continuously monitor the analytes over time. So yeah, so basically the detector can be electrochem, fluorescence. Another student in my lab developed a portable fluorescence detector that we could also integrate into this. So the idea is to make it as versatile as possible. So with that, I hope I showed that you can use microchip electrophoresis with electrochemical detection to look at reactive nitrogen, oxygen species, that you can use multiple electrodes to improve selectivity and also identify specific analytes. Also, some of the work that we've done with using a separations-based approach with LIF detection adds more data than just using the dye itself and making a single measurement. And then just as an aside, the interesting that carnosine seems to be reducing at least nitric oxide production. We're still working on single cell analysis and online microdialysis sampling and trying to make that better for looking at these reactive nitrogen and oxygen species, and hope eventually we'll have portable systems that we can use for either on animal monitoring, but also you could see where you could use the same devices to put it near a patient in the ICU or something, and instead of having to take the sample to the clinical lab or whatever. So there's a lot of people to acknowledge. I think the key ones here are Shamal, Galina, Rachel Saylor, and David Scott, who all worked on this project pretty hard. And actually, and Anton Beribin, who's just been incredible on working on this as well. And Sonia, who's been gone a couple of years, but is now at the University of Washington in Seattle. And then with the cell studies, Dulan, Manjula, and Danushka have been key players on that. And Indica, who's not on here, but should be. And then Chris Culbertson and his group, and also the Shonik group. And so, as you probably noticed, I'm wearing a hat because I was diagnosed with breast cancer, so I have no hair. So I forced my group to take a picture with Lunte research group hats in the middle of July. But this is the current group, and this is Galina, Anton, that have been working on this project, and Indica, who's also been working on it, and then Riley and Danushka, who have been working on the nitric oxide stuff. So with that, I'd be happy to answer any questions. Hope I didn't go over.
No.
Am I right on time?
You're right on time. Thank you, Sue.
Okay, good.
We have a lot of good questions here for you, so I'm gonna just jump right into them.
Okay.
So the first one comes from David.
Oh, yeah, tell him hi.
So, as you described, electrochemical detection has many advantages. Why do you think electrochemical detection is not commonly used or available in commercial instruments? Are there particular challenges in its design? Are there limitations in monitoring large biomolecules?
Yeah, definitely limitations in monitoring large biomolecules, but I think Dave probably knows what the limitations are better than anybody when he was here at KU. I think, for portable sensors, I mean, their problems, I think, are electrode stability. You have to make sure that you're not getting electrode fouling, so it's a little different than an optical detection method where you don't have to worry about fouling. And I think it's really, really good for the analysis of small molecules, but larger. I mean, one of the things I wanted Kelsey to do, but she didn't have time to do, is look at as the peptide gets bigger, your response is going to go down based on, just the ability for it to reach the electrode surface. So I think electrochemical detection is really more uniquely suited for small molecule analytes. And so that's we've been focusing mainly on that. It's been used a lot by different groups to, especially for clinical analysis, because it is small and portable. And there's several companies, Microx, Pinnacle's commercialized a potentiostat that we were working on with them. So there are applications for point of care things, but in terms of big biomolecules, like a lot of people at this conference do, to look at proteins or something, it's not very useful because the kinetics at the electrode are just too slow.
Okay. So the next question comes from Kari. Thank you, Sue, for all the work you do and your group have been doing over the years. So for many years, you've convincingly shown the potential of the chip technology, yet there is still no open platform chip instrument. They're only application specific. Mm-hmm. What are the hiccups?
Actually, I think part of the problem is compatibility. And actually, I was just reviewing a tenure packet of somebody today, and it was sort of interesting because I think part of the problem is, and I'm guilty of this too, we work on a specific application, so we make everything really specific for a specific application instead of thinking broader, like how can we integrate our technology into stuff that's already out there, as opposed to trying to invent everything from scratch, right? So I know there's some groups that are sort of working backwards, like they're looking at how people use 96 well plates or cell culture plates and are trying to integrate the microfluidics into that platform, as opposed to my group and a lot of other groups that fabricate the microfluidic devices and then hope that people will use them for a specific application. So, I mean, I think there's a need for both of them, but I definitely agree that, I mean, because the detector is different for different applications and the chip design is different for different applications, there's not really a one size fits all, but I think we could, if we really thought about it, we could figure out a better way to integrate it better into current instruments, as opposed to trying to sort of push our way into a new way.
Okay. So the next question comes from Layla. Thank you, Susan, for the presentation. How many analyses can you use one microchip? So what's the cost? Do you see variation in the migration times when running samples multiple times on the same microchip?
Yeah, that really depends. The glass chips are more rugged and more reproducible. The all glass chips, like with fluorescence detection, are definitely more reproducible and more rugged than some of the ones we use for eChem, which are hybrid PDMS glass chips. Those tend to change the migration time over time because the PDMS changes. Generally, with the online system, we're getting better. Galina's been able to run it for a couple hours continuously, getting data. We always try and put an internal standard in there for both migration time, but also, especially with electrochemical detection, to make sure that we're not following the electrode and the electrode response is going down. In terms of cost, we make most of these ourselves, so I'd probably not. I probably couldn't even really tell you with all the labor and all that kind of stuff that goes in there. But I know that one of the hopes, right, is if you get a specific design that you can eventually make it so it could be injection molded, and then it could be really cheap. Mm-hmm.
So we have another question. I think this one kind of touches on your last answer. It's really when you design the microchips, how do you build them? And this could be a really long response, I suppose, but we only have a few minutes left. So
yeah,
is there
that's
anything
fine.
you want to add?
Yeah, we do it. Well, right now we make them all in-house. I know there's a bunch of companies where you can have chips made for your chip shop, and there's like 5 or 6 companies. When my students design their chips, they do use some COMSOL modeling sometimes, and we fabricate it. We have an in-house microfab lab. So the way we design is really dependent on the specific application. Is that short enough for you?
Yeah. Thank you. Okay, so another question from Kari. Can you explain a bit how the cell lysis occurs by applying voltage? The electric field strength in our separations is still way below the electric field of ions on the angstrom distance. In addition, we analyze intact virus particles with CE. So is it only the voltage field strength, or is there more to it?
There is, Chris does put some surfactant in there, so I think that helps. But it does, when it enters the separation voltage, then it definitely lyses. So I'm trying to remember. He puts in, I think part of it is there's a surfactant in there that's going to weaken the membrane, and so that helps lyse the cell. It also moves the bigger stuff into the waste reservoir.
Okay. Next question from David Michaels. The microdialysis sampling technology looks really interesting, and I'm sure it would draw a lot of interest from his research colleagues. Have you teamed up with private industry to explore opportunities to further advance this into drug discovery and research?
At times, not recently, but be happy to talk to you for sure. We have done some like drug. The one nice thing about microdialysis, and actually my husband Craig was even more involved in this, is that you can look at a variety of tissues. So you don't just have to look at the brain. You can, like they did work where they looked at tumor versus healthy tissue to look at the differential metabolism in different tissues. You can look at transdermal delivery. We've looked at some, we have done some work with some companies and institutes looking at bioreactors, because it's a really easy way to get stuff out of bioreactors. And that's actually a really easy application of it because the bioreactor doesn't move. So it's a really nice sampling technique that you can either do really fast or really slow. I'd be happy to talk to you about doing something.
Okay, the next question is from Mustafa. Thank you for the wonderful presentation. The fluorescent labels DAF-FM and 6CF, are they detectable only in viable eukaryotic cells, or can they be used in prokaryotic cells and virons also?
Oh, geez. I really don't. I have no idea. I'd have to ask. I'm not sure. Yeah.
Okay.
They go into the cell. They go into macrophages. I know people have used them to look at NO in a variety of cells, and they're viable cells. That's when they're incorporated. But I have no idea about viruses or. It's a really good question. It crosses the cell membrane. I guess I don't know enough about viruses to make that.
Okay. The next question comes from John. In single cell analysis example, you described the use of a syringe infusion wastegate to get rid of unwanted or uninteresting compounds. What properties of the molecule dictate shunting it to the wastegate versus the analysis channel?
So big negatively charged stuff goes to that waste channel, and then one of the problems with trying to connect that single cell analysis method to electrochemical detection, which is something that we want to do, is that we can't put the high voltage end in the detection end, but we can do that with fluorescence. So the waste is all the like DNA, lipids, and some of the proteins go in that direction. And he's got I don't know if it's BSA. He's got some additional sort of solubilizers in there that help move it into that waste reservoir. So in slow moving big molecules are swept away before they're injected.
Okay, and we have one final question from Kari. She likes the lab on the sheep. How do you deal with rinsing the chip and refreshing the buffer between injections?
Yeah, that's a really good question. So when with the example that I showed, he had a fairly good sized reservoir there, so he could run. I think he ran it, I don't know, an hour maybe or so. But we do have a prototype, or we did have a prototype that had like a sort of a peristaltic pump that would fill up the reservoirs, because you're exactly right. You need to make sure that the reservoirs are replenished every time, or flushed every time, every so often. So we did have that built into the design, but we didn't use it in that application that I showed. But that's a really important point, because if you're going to use it for a long time, you have to be able to have be able to keep the buffer reservoirs filled, and also make sure that the waste reservoir doesn't. Well, the waste reservoir you can actually just let drip down the sheet, but the other part you need to make sure it's filled.
Okay, well, thank you, Sue. On behalf of all the attendees of the session, I'd really like to thank you for speaking to us today. I'm sure we all learned a lot. It was a very interesting talk. Thank you. For everybody on the call, we now have a 10-minute break. So the first session on method development and optimization will start in 10 minutes. And that's 9:25 Pacific, 12:25 Eastern time. So we'll see you back in 10 minutes. Thank you.
Yeah, thanks everybody for all your questions. I really appreciate it. Yeah, and for tuning in either really early or really late, over lunch or over breakfast, whatever. All right, thank you.
Thanks.